Phagocytosis is one of the oldest acts in immunology, but it has become highly relevant again in drug discovery. In this assay, THP1 cells were incubated with fluorescent beads together with different antibodies to compare their functionality. In this sample, we used 3 different channels. We used the DAPI channel to identify a nuclear stain. We used the FITC channel to see the fluorescent beads, and we used the Cy5 channel to visualize the membrane. And finally, we have a DIC channel with normal light. First, we identified the nuclei that look like this to identify where individual cells are located. Then we created a membrane mask using the Cy5 channel and used the nuclei to split up cells that are stuck together. Like this. We produced a mask for the fluorescent beads. And finally, we stuck it all together and analyzed the images. And here we can see the results. As you can see here, we have 4 different wells. Each well contains a sample that has been treated with a different antibody. If we zoom in, we can see the individual cells and how they are segmented. First, we wanted to perform a quality control gate using area versus circularity. Here we wanted to gate out cells that only had a high circularity and a normal area. Next, we wanted to compare the efficiency of the different antibodies using a bar chart and plotting the mean FITC signal. And we could immediately see that we have a difference in the intensity between the 4 groups. So while we can see that there is a difference between the groups in the mean FITC intensity, the differences aren't that great and much of it disappears in the background fluorescence. Therefore, we used another parameter to see if this could give us greater differences between the groups. We chose instead to visualize the number of children, which corresponds to the number of beads within each cell. And we can see that using this, we have a similar trend, but the fold change is greater compared to the mean FITC channel. This we can further visualize using a table where we plot both mean FITC signal and also mean number of children per cell. And while we see that there is a very slight difference between the groups in the FITC channel, we have a much larger difference in the number of beads that have been taken up per cell. So this really highlights the difference between microscopy and flow cytometry, where a object per cell analysis is possible.